and I'll go over slide 1. So I want you to notice that you basically have all 5 classes of leukocytes that were discussed in lecture on Monday here. So your job for your lab practical, because we don't really have these slides available to us, so for your lab practical, I'll essentially probably give you like a picture or a handout, and then you get to identify things for me, and then I might ask you for distinguishing features. So that's why we're going to spend some time on this, and then you guys are going to have the opportunity to essentially do the identifications yourselves in lab today. So that's why lab's going to go by a little bit quickly today. The long part, honestly, is this kind of preview lecture and then the checking afterwards, okay? So, if you recall, I'm going to do green here just because. Green one's easier to see. Do you remember that there are 2 classes of leukocytes? No? Okay. One class is called the granulocytes because they have a bunch of granules in the cytoplasm. So they have a bunch of tiny little dots everywhere. And the other class are called your agranulocytes. Everybody here following me still so far? So it just so happens that your 3 granulocytes are actually grouped together here. So I am going to do this, and you guys can see the green okay, right? So the ones at the top are your granulocytes. These ones at the bottom are your agranulocytes. Now, it kind of seems like the neutrophils don't have any granules, but they do. They're just kind of tiny there. But I want you to compare the appearance of the eosinophil and the basophil to the appearance of the lymphocyte and the monocyte cytoplasms. Do you guys see how for these 2 on the right, like you can see a bunch of little dots everywhere, and it's actually most apparent in that basophil. Do you guys see that? Okay, like all of those little black dots that you're seeing floating around, or purple, I guess, those are your granules. Everybody here clear on this? Okay. So, what I'm going to do right now is I'll go through the characteristics of each one. I will start off with the neutrophils first, and let me change the color here. You can't really see the yellow, huh? Purple? Okay, so for the neutrophils, your telltale characteristic for these guys is that they are going to have multi-lobe nuclei. Okay, they're a multi-lobe nuclei. So if you notice in here, okay, notice how you have a nucleus that has a number of lobes, and then at the top left, that's an immature neutrophil. Now, we discussed 2 immature types of neutrophils during the hematopoiesis that we were talking about on Monday. So this one on the left would be considered like a late-stage band cell, early-stage segmented cell. Do you guys see the difference between the immature neutrophil and this guy here, the mature one? Notice how the lobes in the mature one, and notice how they are distinct lobes. I count 1, 2, 3, 4 lobes here, maybe even 5, but 4 at minimum. Does everybody here see that? Whereas in the immature neutrophil, it's basically one band, and then you're kind of starting to form the little divots. Does everybody see the difference between the 2 here? Now for the immature neutrophils, because it is really difficult to tell whether something is mature or immature, I'll tend to not ask you about it. You may have it in one of your slides here, but for the lab practical, I will tend to ask you more about the ones that are already mature and easily distinguishable. Is that clear to everybody here? Okay, but for the neutrophils, look for really well-formed lobes. Thumbs up, thumbs down if we are good on the neutrophils. Okay, great. Awesome. So that's done. Okay. Next thing we're going to talk about are your eosinophils. Eosinophils, their claim to fame is that they will tend to have a bilobe nucleus. for the eosinophils in this one, I have 1 and I have 2 lobes right there. Okay, if you see 3 or more lobes, chances are you're looking at a neutrophil. Okay, which also happens to be the most numerous type of leukocyte that you've got. Everybody clear on this? Can you give me thumbs up if stand if you are? Awesome. Okay. The basophils are probably the easiest to distinguish out of all of these because the basophils have very large amounts of darkly staining granules to the point that sometimes the nucleus is really difficult to see. Okay, so the basophils, when you're looking at them, the basophils, these guys are going to be really easy to tell apart because of the fact that they are going to be releasing these really darkly staining granules, okay? And does anybody remember what chemicals basophils will release?
Histamine.
Histamine, which is, what does histamine do?
It bronchoconstricts.
The opposite.
Oh,
Vasodilation. You guys, basophil will release large amounts of, it'll release 2 main compounds, okay? So basophils, and I will try to write this in green, okay? Basophils will release histamine. Can you guys see the green okay? Which is a vasodilator, and it will also release heparin, which is a blood thinner. Okay, it is an anticoagulator. Okay, so histamine is a vasodilator, heparin is an anticoagulator. Okay, so when you have lots and lots of basophils present, you're probably going to have lots and lots of histamine load, which means that you're probably going to look really puffy, a little bit inflamed. Histamine's really important because histamine, when you induce that vasodilation, it calls more blood to flow to an area. More blood flow equals more leukocytes present, and you can better actively fight an infection. Heparin is an anticoagulator, which also facilitates blood flow. Everybody here good on that? I'm not going to change it yet. I'm not done. I'm not done. Does anybody remember what neutrophils release?
Lysozyme.
Lysozyme. Starts with an L. Ah, no! Almost wrote that. Lysozymes. Okay? So neutrophils will release lysozymes. Okay? Which will help break down any compounds that they'll ingest. Because neutrophils are macrophages. Okay? Well, they're phages. They're phagocytes. They're not macrophages. Monocytes are not macrophages. Okay, they're phagocytic cells, so they need to have something that'll help break stuff down. Does anyone remember what eosinophils release? Kind of sounds like a military rank. You guys have heard of major basic protein?
Oh.
I did say that in lecture, right? Okay. So eosinophils will release a compound known as major basic protein. And if you recall, eosinophils, their claim to fame is that they're going to be apparent in parasitic infections, especially if you have a large parasitic worm load. So that's what eosinophils do. So these are your 3 classes of granulocytes. Any questions on these? Did I explain that clearly enough to everybody? Okay? All right. So now we move on to our agranulocytes, which again, kind of easy to see. So I'm going to start off with our monocytes here. So monocytes are actually really large cells normal nucleus. So if I were to trace out the shape of the nucleus of that particular monocyte, I want you to notice that it is actually kind of kidney shaped here. But remember, monocytes can have either an oval shaped nucleus or a kidney shaped nucleus. But generally speaking, they tend to be really, really big. They tend to be your largest type of leukocyte. Okay. The other ones have been kind of squished, but when they are around, they tend to be the really, really big ones. Does anybody remember what monocytes become when they mature?
Macrophage.
Macrophages. So monocytes will become your macrophages. So I'm kind of giving you a lot, the potential questions that you could be asked is kind of like an addendum to your lab practicals. But remember, all of this information is also relevant to your lecture. And when I say like, oh, I'll ask questions about your lab practicals, have you guys noticed that even though majority of your questions in your lab practicals are ID questions, there may be a few questions where you have, hey, identify this part, and I'll ask you like, oh, how do you know, or what is the function of this part, so on and so forth. So I'm kind of giving you that, I am giving you that information right now, okay? And again, monocytes will become this in green, just to kind of keep they'll mature into macrophages. Lymphocytes, okay, lymphocytes are really easy to identify because lymphocytes will have a large, round nucleus, sometimes with a white ring around it. Okay, that's what lymphocytes have. So I want you to notice the shape of your nucleus for the lymphocyte. Notice how it's basically this nice round shape, okay? And then there sometimes could be a white ring around it. This one actually does have a little white ring. Not super duper apparent, but it is right here. It's not a complete ring, but it is right there, okay? What are your two types of lymphocytes? So from somebody we haven't heard from today. Yeah. No, that's the two classes of white blood cells. There are two types of lymphocytes specifically. Try them. Yeah. B and T cells. So B lymphocytes and T lymphocytes. Do you guys remember what each type of cell is going to be involved in for specific immunity? Which one produces humoral immunity or antibodies, and which one is involved in cell-mediated immunity or close-range combat? Yeah. Yeah, antibodies are humoral, okay? So if you're listening, the two types of lymphocytes are B and T cells, B lymphocytes and T lymphocytes. Most people usually just say B cells or T cells. The B cells produce antibodies. This is what we would call humoral immunity. And again, the reason why it's called humoral immunity is because back in ancient times, the fluids that run throughout your body were called the humors. And your antibodies are circulating proteins. That's why it's called humoral immunity, okay? And antibodies, if you recall, this is what I would liken to far-range combat, right? These would be like your throwing stars or your arrows or your bullets, okay? Because you don't necessarily have to be nearby to your target for you to induce some sort of damage, okay? By the way, antibodies, when they bind their targets, okay, they'll render the targets harmless, but the other thing that they'll do is they'll cause clumping. They will cause clumping to help slow down the thing that needs to be destroyed. When they are clumped and they're slowed down, that gives the other types of white blood cells, including your macrophages, your neutrophils, your natural killer cells, and your T lymphocytes, it gives them the time to show up and do what they need to do, because those cells that I just mentioned, they have to be in close contact with their target cell for them to induce damage, okay? So the T cells that we are talking about, that's what we would call cell-mediated immunity, because you have to be really close to your target cell to induce some sort of damage. So the analogy that I'm gonna give you guys for that is if you were, you know, like a ninja or something, or I don't know, some sort of fighter in like a wushu epic. I watch a lot of wushu epics with my husband, okay? An anime. So if you were a ninja and the weapon that you were given is a sword or a nunchuck, or your fists, right? You can't induce damage to your opponent unless you are close by, right? A sword can only go as far as the length of that sword and the reach of your arm. Whereas if you have an arrow or a bullet, you could be meters and meters away and still inflict damage, okay? Yeah,
So what are perforins?
what are what?
Perforins?
Perforins. So perforins are special chemicals that T cells will release, okay? That forms pores on the target cell, causing that cell to lyse. But for perforins to work, the T cell has to be nearby, okay? So if you poke a hole using a sword, right, you have to be close by. So for the T cells to poke a hole, they need to put perforins on their target cell. But for them to be able to put the perforins on the target cell, they also have to be close by. So they're poking holes. The perforins would be kind of like the swords of the T cells. Let me write that down for. Cell-mediated or cellular immunity. I should write that down. Any questions on this particular slide, everybody? Any questions on that one? All good? Okay. So, we can all do A together. Which of the types of leukocytes do you think A is? This will be the only one that I'll do for you.
Basophil.
How do you know that it's a basophil?
Because all I see is the dark granulated dots and not a clear nucleus.
Right. Okay, so does everybody agree that this is a basophil? Challenge you? I'm not gonna do that right now. Don't challenge yourselves. Okay. So what I'm doing right now is I'm gonna skip all the way out to slide number 18. So that means that 2 through 17, the ones with the labels at the very top, those are the ones that you all get to work on. I'll give you about 25 minutes to do that today. So like I said, my goal is to be done by like 10 o'clock today, which I think we can definitely do. See, I told you this is why like this lab is fun. You guys get to work on it together, and it's short. fairly straightforward too, okay? So the first type of disease that we're gonna talk about is called sickle cell anemia. So sickle cell anemia is a disease that tends to be more prevalent in people of African or African American descent. No, don't read into this, okay? There are just some diseases that tend to be more closely associated with certain ethnicities. So sickle cell anemia tends to be more prevalent in people of African descent, African or African American descent. Thalassemia and hypertension tends to be more closely associated with people of Asian descent. Okay, Tay-Sachs disease, more closely associated with people who are Jewish, and then cystic fibrosis tends to be more closely associated with individuals who are Caucasian. Okay, so with sickle cell anemia, this is due to a single nucleotide mutation in hemoglobin. Okay, that single nucleotide mutation, so that means like literally if you're looking at DNA, it is a change in a single letter. And that change in one single letter causes a change in one amino acid that comprises your hemoglobin chains. Well, that one amino acid change causes hemoglobin to not form properly. This slide that you're looking at is somebody who is in what you would call sickle cell anemia crisis. Okay, so normal hemoglobin, you guys, and you should have learned this in your lecture videos, okay? Normal hemoglobin is comprised of 4 chains, 2 alphas and 2 betas. And when you put those 4 chains together, they form a globular unit, so it's kind of round, okay? That is the best shape for maximizing efficiency of oxygen transport. When you have the mutation that causes sickle cell anemia, your hemoglobin does not end up forming these nice globes. Instead, they will kind of form these kind of curved lines that will end up, when they come together, they'll end up kind of hooking like this. So I want you guys to think of kind of like a barrel of monkeys. Have you guys ever seen that game? So you have like a little hook, and then you hook another one, and then another one hooks onto it. So that's what happens in sickle cell anemia. So instead of having nice globular chains, okay, you'll end up with a linear chain that elongates your cells. So remember, normal red blood cells are supposed to have what kind of shape?
Biconcave.
Biconcave disc. They are kind of like a jelly donut with a middle squished in, but the technical term for this is called a biconcave disc. That biconcave disc is the most efficient shape for transporting oxygen. If you have sickle cell anemia, your cells tend to be sickle shaped. They look kind of like a bent half moon, okay? Or a crescent moon, I should say. So that abnormal hemoglobin is what we would call hemoglobin SS. So all of the important parts for the description have been underlined for you, okay? So it's a sickle shape, okay? Well, the problem with the sickle shape is that they also tend to, like barrel of monkeys, they'll tend to do what? Stick. They'll tend to stick to one another and form clumps. Well, those clumps prevent blood flow. They will clog capillaries, which means you will not get as much oxygen transport. So individuals who have sickle cell anemia, they have trouble transporting oxygen, and when you have trouble transporting oxygen and you're clogging your capillaries, they tend to be in a lot of pain. And it is pain that is spread throughout the body. Any questions on this? Okay. Yeah.
It's not really related, but what causes like a blood clot inside your body?
A blood clot,
Yeah.
thrombocytes and fibrinogen, technically fibrin. So you have a bunch of clotting factors. So I want you guys to imagine, okay? You have a bunch of I want you to think of your thrombocytes or your platelets is basically tiny little pieces of cloth. Okay. And have you guys ever made like paper mache or anything like that? Like in grade school? Okay. And like, did you make your own paper? No. So when you make paper mache, right, you have little bits of pieces in water. That's kind of like what your platelets are. If you want to clump them together, do you guys notice how you normally have to like sieve it first? And then they'll start coming together. So the thrombocytes are kind of like the paper. Fibrin, okay, and thrombin are your mesh that helps catch the thrombocytes so that they form a plug. Because otherwise, the thrombocytes will just flow throughout the body, okay? So that's how clotting works, okay? It has nothing to do with the red blood cells, because red blood cells, their job is to flow. Everyone clear? Yes? Okay. Oh, what did I just do? Oh, oops. There it is. Okay. Next up, you have a disease called iron deficiency anemia. It sounds exactly like what it is, okay? And iron deficiency anemia, you can tell it from normal hemoglobin conditions because the red blood cells tend to be very small. So if your blood cells are normally supposed to be about this large, they're gonna be in iron deficiency anemia only about this big, about half to 2/3 of this size. So they're very, very small. Another thing that you're going to notice is that with iron deficiency anemia, do you guys notice how there is a big white circle in the middle of your erythrocytes? This is what you would call the zone of pallor. The zone of pallor tends to be a lot larger compared to these guys right here. Notice how they do have a zone of pallor, but the zone of pallor tends to be very small. That zone of pallor is where your biconcave disc kind of pinch in towards the middle, so it does tend to be a little bit thinner and allows more light to pass through under a microscope. But when you have iron deficiency anemia, that zone of pallor tends to be a lot larger. Why? Because you need iron to make what protein? What is the main iron that allows red blood cells or erythrocytes to transport oxygen? I literally just talked about it in the previous slide. Hemoglobin. In the very centers of your hemoglobin proteins is iron. So if you do not have enough iron, okay, you're not going to have as much functional hemoglobin, which means you're not going to be as red. And that's why you have a large zone of pallor here. Okay, this is what we would call a hypochromic syndrome. Chromic means color. What does hypo mean? Below or less. So something that shows less color. This is also what you would call a microcytic disease. Microcytic. What does cyte mean? Cell. What does micro mean? Small. So notice how iron deficiency anemia shows both of these characteristics. You have a large central zone of pallor that doesn't have as much color. That's the hypochromic portion. And then you also have cells that are smaller than normal. That's the microcytic portion. Thumbs up, thumbs down if I explain this one clearly enough to all of you. Okay? Yes? Okay. Up next, you have a different kind of anemia. Okay, this is called microangiopathic hemolytic anemia. Okay, and for this one of your telltale signs is that you have a bunch of fragmented red blood cells. Okay, so I want you to notice what is in that name. Notice how it says hemolytic. What does hemo mean? Blood. What does lytic mean, or lytic? To burst, to break apart, okay? So this is MAHA, okay? So notice how you have a bunch of tiny little fragments here. So I have these guys right there, okay? So these fragments are what you would call schistocytes or schistocytes, okay? That's what the fragments are called. Now some of these fragments, some of these schistocytes have a particular shape. These are what we would call your helmet cells. I'm gonna point one out right here. This is a helmet cell. And I want to, and when they're talking helmet cells, okay, they're technically talking about the ones from like the military ones. But if you have a kid or if you ride a bike, not a bike at the gym, okay, you guys, but like a bike that actually takes you places, not a stationary one, okay. Pelotons don't count because you're like staying there even though you have like your coach and you can, that's the thing with Peloton, right? Like, can you do that thing on a Peloton? Yeah. Or you're like watching the screen and it's like you're on a road, but really you're still in the same room. Okay. These are actually based on military helmets. So in military helmets, they're called helmet cells because your helmets are typically shaped like this. And then you have the straps that are hanging down for the bands that go around your chin. Okay? So look at the helmet cell schistocytes here. Notice how they have things that are kind of popping off on the sides. Right here. And here's another helmet cell right here. This one would technically also be considered a helmet cell even though it has like 3 little bumps instead of 2, but that's still technically considered a helmet cell. Any questions on this one? So for this, if you see red blood cell fragments, schistocytes, chances are you are looking at microangiopathic hemolytic anemia. Any questions on this one, everybody? We have about 3 more to go through, okay? This next one is called hemoglobin sickle cell disease. This is not the same as hemoglobin as sickle cell anemia. Okay, this is hemoglobin sickle cell disease. This also affects hemoglobin, however, it is due to a different mutation and it produces a different type of hemoglobin, okay, called hemoglobin S and C, okay? This is not anywhere near as common as sickle cell anemia, okay? But what I want you guys to look at here for hemoglobin sickle cell disease is that you do not have fragments. However, what you do have are rectangular shaped, rectangular red blood cells. So the thing that I've circled here, the one that looks like a bar, notice how there's no central zone of pallor in that bar. That lets you know that you have hemoglobin sickle cell disease here, okay? Another thing that you can see here, okay, is that, so here's what I want you guys to know, okay? So hemoglobin S, okay, hemoglobin S and hemoglobin SS, very, very similar to one another, but there's also another type of abnormal hemoglobin called hemoglobin C. Your cells may sickle here, it can kind of form that kind of like half moon shape almost, that crescent shape. However, what is more prevalent for hemoglobin sickle cell disease are target cells. So I want you to look at these 2 cells right here. On normal circumstances, you should have just a central zone of power, okay? A small central zone of power. What do you notice here?
Another one.
But it's not a central zone of power. Notice how it's a dark spot in the middle. So this is what gives you your target cell appearance. Again, that's a telltale characteristic of hemoglobin SC disease, okay? So that's your target cell. And this is due to the fact that hemoglobin C will clump up, but when they clump up, they'll tend to stay in the center of that cell. It's because of the way that they tangle up together. Whereas hemoglobin SS, which you find in sickle cell anemia, when they tangle up together, they tend to form fibers, which elongates the shape of your red blood cells. Any questions on this one? All good? Thumbs up, thumbs down for me? Okay, 2 more, you guys. Okay. Next one, you have something called megaloblastic anemia. Okay, megaloblastic anemia. Hey guys, what does blast mean?
Immature.
A baby, mature, immature.
Immature.
It's an immature cell, okay? What does mega mean? Large, okay? So in megaloblastic anemias, these are so called because what you end up having are these really, really large, immature granulocytic cells. They'll tend to be neutrophils, because neutrophils are your most numerous type of leukocytes. But I also want you to notice something about the lobe of this, not the lobe, the nucleus of this neutrophil. How many lobes do you see?
Many, multiple.
A lot. You're looking at about 6 to 8 lobes instead of normally having 3 to 5 lobes or 3 to 4 lobes. So you have a hypersegmented or hyperlobe neutrophil. That is your telltale sign for megaloblastic anemia, okay? Your red blood cells are also larger than normal. This is now what we would call a macrocytic disease. Remember earlier we had something that was microcytic, where the red blood cells are smaller than normal. In here, you have macrocytic, larger red blood cells. This is due to a folate deficiency. Folate is the same thing as vitamin B12. But I want you guys to look at your red blood cells. Notice how the red blood cells are essentially showing the right shape. They do have a central zone of pallor, but notice how that central zone of pallor is not very big relative to the cell. It doesn't take up the majority of the volume of that cell. Everybody here following me still? Yes? But what you do have, and they are pretty big relative to the size of that neutrophil, okay? These red blood cells are really, really large compared to that neutrophil here. But again, the best thing for you to look at here when you're looking at megaloblastic anemia is to look for neutrophils that have way too many lobes in their nucleus. Any questions on this one? All good? Okay, and again, when you're studying, what should you be paying attention to in these descriptions?
Underlined.
What?
Underlined.
Anything that's underlined, because that literally lets you know the parts that are super duper important, right? And it tells you the characteristics too. And then our last bit here, you have something called hereditary spherocytosis. Okay. These red blood cells tend to be very small. And you notice how I've been talking about this thing called the zone of pallor? Okay, what do you notice about these guys? Do they have it?
No.
They do not have it. It's just a uniform color all throughout. Okay, so this is different than the other ones. The reason why they do not have that central zone of pallor, and remember I told you that central zone of pallor is indicative of that biconcave disc shape. Okay, so when you have that biconcave disc, remember you're pinching in the middle, which means that that middle portion is a little bit thinner. So when you're looking at red blood cells under a microscope, that thin portion should allow a little bit more light through. That's why it looks lighter, okay? The reason why this is called hereditary spherocytosis is because there is an issue in the red blood cells where you do not make a protein called spectrin. If it's underlined or bolded, that's really important for you, spectrin. Spectrin is a cytoskeletal component of erythrocytes. These spectrin proteins help induce that biconcave shape. If you do not have spectrin, the cell tends to be unstable, so they're gonna want to form a shape that provides the most amount of stability, and that shape just so happens to be a ball or a sphere. So if you look at the name of this disease, hereditary spherocytosis, what does cyte mean?
So.
And sphero means
Running.
sphere, balls. So you have red blood cells that are shaped like little balls. And when you are a little ball, that means that your volume is equal throughout, right? There isn't a particular place in that red blood cell that's going to be thinner than the others, and that's why you do not have your central zone of power.
Ball cells.
Huh?
Ball cells.
Ball cells. That's literally what spherocytosis means. You have cells that are shaped like tiny little balls. Any questions on this one? Okay, and again, part of the reason why that is again, you're trying to reduce instability of the membrane. And the instability is gonna be reduced by a particular shape, and that shape happens to be a sphere. Because a sphere equally distributes tension equally throughout that shape, throughout the cell. Any questions on this one? Is there anything else you would like for me to clarify today? No? So at this point, I finished the parts that I am gonna go through today until we come back together and go through the rest of the slides. So what I'd like for you guys to do right now, you can stop recording me.